Good evening, Neha. Good evening, Paramjeet. Okay, let us start our topic that is proteins. Is my voice clear? Okay. So, with the note, proteins are the most abundant biomolecules. These are the most abundant biomolecules, and Rubisco. Heard of this enzyme? It is a protein that is the most abundant protein in the whole like biomolecules, and it is involved in the carbon fixation in the plants. Proteins can be like as large as it consists of 27,000 amino acid residues, means it can consist of around 27,000 amino acid residues, and it can as small as it can only consist of two amino acid residues. The example is aspartate, aspartame. It is an artificial sweetener. It is an artificial sweetener. It is made up of two amino acid residues that are aspartate and phenylalanine. And the one I was talking about that consists of 27,000 amino acid residues is a titin named protein. This is important. Rubisco is asked many times in exams. Okay, what I have to repeat, I'm like starting from the beginning. Proteins, these are the most abundant biomolecules. The categories that we discussed in the previous class, apart from carbohydrate, lipids, and nucleic acids, proteins are the most abundant biomolecules. Rubisco, it is an enzyme. It is involved in carbon fixation. It is the most abundant enzyme and it is in proteinaceous in nature. It is the most abundant biomolecule. Then I was saying proteins can be as large as it can consist of 27,000 amino acid residues, and it can be as small as it consists of only two amino acid residues. The example is aspartame. It is used as artificial sweetener. It consists of two amino acids, aspartame and phenylalanine. Is it clear now? Is it clear? As you all know, amino acids are just the polymers of amino acids, and they consist of peptide bond. Is it fine with these two statements? Even they consist of wide range of functional groups. Name the different functional groups that you have seen in the amino acids. For example, the COH carboxyl group, the amide group, even the hydroxyl group, the amino group. So different amino acids consist of different functional group, but two of which are common, that are carboxyl and amino. These two functional groups are common in every amino acids. So we can say that every amino acid consists of carbon, hydrogen, nitrogen, and oxygen. Is this clear? Okay, fine. So the next property is the third one. They interact with each other and other biomolecules to form complex assemblies. Proteins interact with even the proteins, as well as they interact with other biomolecules. On the basis of composition, proteins can be divided into two categories. Okay, I said amino acids consist of different functional group. Yes, okay, fine, no issue. They consist of different functional groups. Carboxyl group, hydroxyl, amine, amide. These are the various functional groups, but in these two are common in every amino acid. Now, I said protein interact with proteins and as well as with other biomolecules. And based on this type of composition, the proteins can be divided into two categories. The first one is simple proteins. Means they will consist of only the amino acid residues, and the another category is conjugated proteins. Conjugated proteins consist of the proteinaceous part as well as the known protein content, and that known protein content, it is known as prosthetic group. It is known as prosthetic group. The known protein content can be the metal ions or the carbohydrate or even the phosphate group, and depending on these particular like that. For example, glycoprotein, when it consists of protein plus carbohydrate. Phosphoprotein, protein plus phosphate group. Protein, when it consists of protein as well as heme group. Are the examples clear? Okay, I was saying the proteins can be of two types. The first one is simple proteins, means they will consist of only the amino residues part. And the other one is the conjugated proteins. They will consist of the protein part as well as the non-protein part. The non-protein part can be carbohydrate, phosphate group, or the heme group. And there are many categories. These are the common examples. And the conjugated protein without this prosthetic group is known as apoprotein. See, when the complete thing is present, protein plus non-protein part, it is holoprotein. It is termed as holoprotein. And when this means holoprotein minus prosthetic group equals to apoprotein. Is it clear? The same thing is in case of enzymes also. We will study these things there also. Holoenzyme, prostatic group, apoenzyme, as the enzymes are also proteinaceous in nature. Is it clear? Protein is the conjugated protein that consists of both the proteinaceous part and the non-content. For example, when I say glycoprotein is the holoprotein, it consists of protein that is the carbohydrate. Audible now? Am I audible now? Yes. Simple can be considered as holoprotein till the part they are complete. They don't like any residues. They can be termed as holoproteins. Next category we have is it audible now? Some proteins are rigid while others are flexible. What does this mean? Rigid and flexible means they do not change their structural organization. They remain same as it is. And that is why they are involved in the structural elements. The function of rigid proteins is they are involved in the structural elements in the cytoskeleton. While in case of proteins that are flexible, they act as hinges, springs, and levers. Example of flexible proteins are lactoferrin. This is a protein that will change its structural organization when it is bound to the iron component. When the iron is absent, the structural composition or the shape of that protein is different. But when it attaches to the iron part, it changes its shape. Is it clear? Is it clear? Livers. Lactoferrin is a protein that consists of an iron part. But when the iron is absent, the shape of that protein is different. But when the protein attaches to the iron part, it changes its shape. Flexible means they can alter their shape. The like if I tell simply about the hinges, springs, and levers, they act as the connecting links. Now, the next topic that we have is how to find out the molecular weight of a peptide. See, this is one amino acid. This is some another amino acid. Here, loss of one water molecule occurs and it leads to the formation of peptide bond. Is it clear? So what we have to notice is when there are two amino acids involved, when two amino acids are involved in formation of a peptide bond to form a dipeptide, then it leads to the like it leads to the leaving of a water molecule. And the molecular weight of water molecule is 18. Is it fine? When I say two amino acid residues combined, it leads to the leaving of one water molecules. When I say 10 amino acids are linked in a peptide, how many water molecules are removed? Okay, yes. So the basic part of this type of numerical lies here only. Number of water molecules removed are n minus 1, and here is the number of amino acids. Consider these to be amino acids forming a peptide. Here one water molecule is removed, here one water molecule is removed. Is it fine? So I'm giving a numerical. Try to solve it. The molecular formula for glycine is C2H5O2N. Then what will be the molecular formula for linear oligomer made by linking 10 glycine molecules? This is molecular formula for one glycine molecule or amino acid Then a oligomer is formed by linking 10 amino acids. We have to calculate the molecular formula. See what we will do? We will multiply by 10 and subtract 9H2O. Is it clear? Okay. Now I'm giving another numerical. Uh, I will ask Gaurav sir. I'm recording the lecture, but I will ask Gaurav sir. See if the molecular mass of an amino acid is 150 Daltons, what will be the molecular mass of tripeptide? This is the molecular mass of one amino acid. What will be the molecular mass of tripeptide? No, Gunjan, wrong. No, wrong. अभी तक सही आंसर नहीं आया है. पल्लवी, wrong. No, Ikra. Yes, yes, Gunjan. This one is right. 150 into 3 minus 18 into 3. The answer is 414. You don't rush into things. This question was asked in GATE, and even this question was also asked in the same exam. दोनों क्वेश्चन एक ही एग्जाम में एक ही साल पूछे गए हैं. तो अमाइनो एसिड्स और प्रोटीन से बहुत सारे क्वेश्चंस आते हैं, और ये पहले न्यूमेरिकल्स हैं जहां से बहुत ज्यादा क्वेश्चंस आते हैं. तो डोंट रश इनटू द थिंग्स. बहुत इजी है, बट ध्यान से करने हैं. ट्राइपेप्टाइड है, इट मींस 150 मल्टीप्लाई बाई 3, बट वाटर मॉलिक्यूल्स भी तो रिलीज हो रहे हैं. माइनस कितने रिलीज होंगे? ट्राइपेप्टाइड है तो इज इट क्लियर? दो रिलीज होंगे. Okay, I'm giving another numerical. If the molecular mass of a amino acid is 70, it forms dipeptide Am I audible? Am I audible now? Okay. So did you solve that numerical? What would be the molecular mass of a dipeptide? Okay, fine. So you got that from one side. So the average molecular mass of an amino acid residue, average molecular mass of amino acid is considered to be 110 Dalton. Many times questions are asked like this. The approximate length of a protein is 55 kilodalton. How many amino acid residues will it contain? Do I have to repeat or write it down? This was the question. The length of 55 kilodalton protein will be, means the question is asking how many amino acid residues will the protein consist of? Yes, right. See, this means amino acid residue here means the molecular weight of water is already subtracted. We don't have to look into that here. So directly we will divide with this and our answer will be 500 amino acid residues. I told you the average molecular mass of an amino acid residue is 110 Dalton, and the question was the length of 55 kilodalton protein will be, and there will be options given. Yeah, and you have to calculate the amino acid length. Okay, I'm giving another question that was asked in IISc. The closest estimate for the number of amino acid residues in a protein with molecular weight 85 kilodalton. This question was asked in
मैं चुप रहा ना ईयरफोन में घुस घुस के आवाज आती
Okay, sir. The question was the closest, I'm writing, the closest estimate for number of amino acid is it fine now? We have discussed three types of questions. I'm repeating it. See, the first type was in the molecular formula type, means the composition of carbon, hydrogen, oxygen, and nitrogen was given. And then we were asked about the molecular formula of a peptide and the residues were given. So we will have to multiply with the n number and subtract with n minus 1 H2O. Is this one clear? Is this category clear? In the next category, we have given the molecular weight of single amino acid. Molecular weight of single amino acid was given, and we have to calculate the like molecular weight of the protein consisting of the n number of amino acid residues. Similarly, we will use the same formula. And in the third category, we have given the molecular weight of protein and we have to calculate the number of amino acid residues. In this, we are not considering water molecule separately because in 1110, we have included the molecular weight of water also. Is it fine? Sir, in tripeptide, how many molecules? 3 amino acid residues will be there. This will be the tripeptide. This was the numerical part of like proteins. Now we will see what amino acid residues are and their structures too. So as we start amino acids, the first thing is there are about 900 amino acids till now that are known. Don't say 20 or 22 amino acids are known. No, there are around 900 amino acids that are known till now. I don't remember the question, but you can see the recording and any of you can post the question in the chat box. I was talking about amino acids and there are 20 standard amino acids. Means their genetic information is present in our DNA and they are coded by genetic codes. The answer was 772 residues. Yes, answer was 22 residues. Is it fine now? Now we have some proteogenic amino acids too. Means these amino acids are incorporated in proteins during protein synthesis. And when we say about basic structure, during that you have seen the basic structure of the amino acids. This is the basic structure. Based on this R group, we have different amino acids, but the classification is based on the charge. The classification of amino acids is based on the charge, what charge they carry. And for writing amino acids, we have three-letter designation and a single letter code for different amino acids, which we have to remember. In exams, generally, three-letter code is given. Nobody will like say glycine and write full glycine and lysine, but they will write the three-letter code. And sometimes single letter codes are even asked in the exams. This is somehow cramming part. We will do one or two categories in a single day and carry on other like other topics also. Is it fine with you guys? So when we talk about classification, we have with hydrophobic side chains, the amino acids that consist of hydrophobic side chains. That consist of aromatic side chains. Than the hydrophilic side chains. The hydrophilic one is further divided into two categories. The first one is polar, but it is uncharged. And the other one is ionizable side chains. Today we will see the aromatic amino acids. Okay? As we have discussed the numerical part, even that is too much. So we will discuss only the aromatic amino acids. The mnemonic learning this is WIF. WYF. These are the single letter codes. Tryptophan, tyrosine, and phenylalanine. So let us see these one by one. The first one is tryptophan. Three-letter code for tryptophan is TRP, and the single letter is W. There is a neurotransmitter that is synthesized from tryptophan, and it is generally asked, and that neurotransmitter is serotonin. And let me draw the structure first. This one is important, serotonin. It is a neurotransmitter that is synthesized from tryptophan. There is a vitamin that is niacin that is synthesized from tryptophan. And for botany students, this tryptophan is converted to auxins during apoptosis. Tryptophan is converted into auxin where it occurs when sieve tube elements they undergo apoptosis. The amino acid tryptophan, it is converted into auxin. And in general, I'm telling you, tryptophan, phenylalanine, and tyrosine, they are involved in protein detection at 280 nanometer. Even nucleic acids, we detect using UV spectroscopy proteins as well as nucleic acids. The wavelength for nucleic acid detection is 260 nanometer, and for protein detection is 280 nanometer. And we are able to detect proteins due to the presence of aromatic amino acids that are WYF, and other lambda max is 280 nanometer in case of protein. Even this question was asked in GAT. Okay, I'm repeating again. Okay, Tamanna. I said protein detection using UV spectroscopy. It is done by the help of aromatic amino acids, and the wavelength that is used is 280 nanometer. And the detection of proteins is only possible due to the presence of aromatic amino acids. And the same technique, that is UV spectroscopy, is also used for detection of nucleic acids, but the wavelength here is 260. Many students got confused between 280 and 260. Just remember, nucleic acid consists of DNA or RNA, or simply take N, nucleic N for nucleic acids, and here for P for proteins. What comes first is N. So what is smaller wavelength? That is 260, and this is 280. Many people get confused here. Just remember, for proteins, we have wavelength 280 nanometer, and for nucleic acids, we have 260 nanometer. Is it fine? Next amino acid that we have is tyrosine. Three-letter code is TYR, and single letter code is Y. See, when you will do signal transduction in cell biology, you will hear this name again and again, tyrosine kinases, tyrosine kinases. They function as receiver of phosphate groups that are transferred by protein kinases, and they form tyrosine kinases that are involved in signal transduction process. Tyrosine, for botany students, tyrosine residues plays important role in photosynthesis in chloroplast. In photosystem 2, it acts as electron donor. I'm writing it. It acts as electron donor. Photosystem 2, chloroplast. See, the structures are not important. You don't have to cram the structures, but the points are important. And the single letter codes and like for aromatic amino acids, the wavelengths, these all things are very important. Is it clear? Next amino acid we have is phenylalanine. The three-letter code is PHE and single letter code is F. See, अब तक जो भी हमने तीनों अमाइनो एसिड देखे हैं जनरली जो फर्स्ट थ्री लेटर्स थे वो ही उनके थ्री लेटर कोड्स में आ रहे हैं, बट सबके ऐसे नहीं होते, कुछ कुछ में चेंजेस होते हैं, वो मैं बता दूंगा कहाँ चेंजेस होते हैं. Phenylalanine, it is very important amino acid because it acts as precursor for many biomolecules. First of all, it acts as precursor for tyrosine, means it is involved in the synthesis of tyrosine, then dopamine, norepinephrine, epinephrine, and melanin. It is involved in the synthesis of tyrosine, dopamine, norepinephrine, epinephrine, and melanin. Melanin is a skin pigment. ये जो भी पॉइंट्स लिखे हैं ये एक एक बार सब में पूछे गए हैं, and this is commonly asked. And phenylalanine, due to its hydrophobicity, it is generally buried inside the protein. And during the folding of proteins, the phenylalanine, it helps in the stability of structure. During folding, it helps in stability I am writing all the points. These are the 3 points. The first one is it acts as precursor for the synthesis of tyrosine amino acid, dopamine, norepinephrine, epinephrine, and melanin. Melanin is a skin pigment. And during the folding of what? Folding of proteins, due to its aromatic ring, it helps in the stability during the folding process. Again, due to its hydrophobicity, it is buried inside the protein, means the core is made by hydrophobic amino acids and phenylalanine is one of these. Now I'm making the structure here. तो फिनाइलएलेनिन और टायरोसिन में डिफरेंस क्या है? यहाँ पर OH था टायरोसिन में, फिनाइलएलेनिन में नहीं है. इज इट फाइन? ये क्लियर है? कुछ पॉइंट्स जो कि अमाइनो एसिड के स्ट्रक्चर्स रिलेटेड है वो एज इट इज याद करने ही है, उनका कोई शॉर्टकट नहीं है. तो मैं उन पॉइंट्स को ज्यादा एक दिन में डिस्कस नहीं करूंगा. somebody wrote CHO. जो पैरा पोजीशन है उस पर OH प्रेजेंट था टायरोसिन में. इज इट फाइन? कोई डाउट्स है आज की क्लास के? आर देयर एनी डाउट्स? Even you can ask me in the group itself or you can DM me. So thank you everyone. That's all for today.